All right, welcome everybody. This is the fourth chapter in A&P, third edition, McKinley, O'Loughlin Middle, and this deals with um, cell biology, which is the smallest unit of living matter. Okay, so the objectives are listed here. Okay, cytology is an example of microscopic anatomy and physiology. Okay, you have to use a microscope in order to see the cells that make up the human body because they're too tiny to visualize with the naked eye. We generally measure these in micrometers, which is 1/10,000th of a centimeter. So these are extremely small units. Um, the cells are the smallest unit of living matter because they can do everything we associate with life, right? They grow, they can reproduce, they can respond to stimuli, they can metabolize, they have barriers, they have the ability to excrete waste, and they have the ability to change over time. All of these are considered processes that are germane to living things. The microscope, along with staining techniques, is critical to visualize these building blocks in order to describe their features and thus infer their function. Different kinds of microscopes. The light microscope is the one we're going to be using in this course, and it uses visible light in order to visualize the specimen. So we're in a very specific wavelength in the electromagnetic spectrum that allows us to pick up objects as small as a mitochondria, but nothing really much tinier than that. It passes light through a stained specimen in order to visualize the structures because normally when we're talking about most tissues, they don't come with structures that bear their own pigmentation. So we have to stain them somehow in order to see them. Electron microscopes are much more expensive pieces of equipment. They cost between $1 million, $2 million on average, and they use a beam of electrons that hit a fixed specimen that's been um, dehydrated and coated in tungsten in order to visualize structures that are as small as viruses. And the reason for that is that you have to have the tungsten in order to deflect these small wavelength beams of electrons. And the use of something that has such a small wavelength means that tiny objects can deflect the beam and thus create an image that we can analyze on a detector or on a printout. Two different types are scanning and transmission. So you can see some examples. Light microscope there on the left, scanning in the middle, and transmission on the right. So you get a sort of a three-dimensional image with the scanning, sort of a 2D image with transmission, and obviously a 2D image with the light microscope. An important point is that because you have to dehydrate the specimen and coat it with tungsten, sometimes you create structures in preparation for electron microscopy that aren't present in the actual object, and these are called artifacts. So we have to be on the lookout for something that might not represent the structure in the actual living specimen. Cells are highly variable, right? A muscle cell, a skin cell, a connective tissue cell look very different from each other. They vary in size, as you can see here. Um, most are microscopic. There are some exceptions. Nerve cells can be quite long, a foot long or longer. Skeletal muscle cells the same. Um, their shapes will vary. Their arrangement will vary. And their physiology is going to vary. The reason for this is that the cells have genetic material in their nucleus that directs their activity by coding for the proteins that they use to carry out life processes. That genetic material is used differently in different cell types. Okay, You could think of a muscle cell as reading from the muscle cell handbook of its DNA, a nerve cell as reading from the nerve cell handbook, and so on. The information in the nucleus in these different cell types has been modified so that only the important information for the cell to operate is available for transcription and translation. You can see the range of cell sizes here. So we're going to be looking at stuff that is in the yellow range, right? So 100 microns down to a micron. We can't pick up stuff below that, again, because the wavelength of visible light is too big to deflect objects that are so tiny. You can see some different cell shapes here. Again, notice that the arrangement, the shape, the location are different, and that generally reflects the function of the cell. Okay? Again, think in terms of the DNA, right? All these cells, save the red blood cells pictured here, have the same DNA in them, but the information, again, has been chemically modified in each of these cell types in order to only allow the relevant genes to be used to direct the activities of the cell. Common features: the plasma membrane is the outer phospholipid bilayer, and sometimes you'll see extensions such as cilia and flagella, which are microtubule-based structures that use ATP for fuel and promote movement. Cilia promotes movement of materials across the surface of a cell, while the flagella promotes movement of the cell itself. Microvilli are essentially extensions of the cell membrane designed to increase surface area, and their structure is maintained by microfilaments. The nucleus is the largest structure in the cell. It contains genetic material, which is chromatin, which is DNA complexed with histones and non-histone proteins. And the nucleoplasm is just the matrix in which the DNA is suspended. The cytoplasm is just everything between the cell membrane and the nuclear membrane, and it contains cytoskeletal components, waste products, electrolytes, water, and organelles, be they membranous or non-membranous. Okay, the non-membranous organelles include the centrioles and centrosomes, and the cytoskeletal components and the ribosomes, while the membranous organelles are all the remaining structures. So that would be the lysosomes, peroxisomes, rough and smooth endoplasmic reticulum, the mitochondria, okay, and the Golgi apparatus, as well as the transport and the secretory vesicles. The components of the cytosol, again, listed here, the organelles listed there, okay. And again, the difference between the two, right? Membranous organelles are surrounded by a phospholipid bilayer, while non-membranous are not, okay? The purpose of having organelles is to isolate chemical reactions in a physical space so that they're more effective, so they do their job better, because usually a series of chemical reactions is ultimately used to direct cell activity. We also want to isolate chemical reactions that interfere with each other so that they don't work against each other inside the cell. So if we were to say have a set of chemical reactions that synthesized a particular molecule and another set that tore that same molecule apart, they would be located in different organelles so that they wouldn't run at cross purposes. Inclusions are simply regions of temporary storage inside the cell, either useful material such as fuel like glycogen or triglycerides, or materials such as pigments or other waste products, sometimes crystals, okay, depending on the cell type. You can see an example of the cell here. We'll go through each of these structures as we wander through. You can see the membrane, the cytosol, the membranous organelles, the nucleus, and the chromatin, okay? And we'll get into each of the components one by one. Cells are critical because they maintain their shape and integrity because of the cell membrane and the cytoskeletal components and the cell-cell junctions. They get nutrients and form chemical building blocks from the extracellular environment so that they can continue to function. They dispose of wastes, Many are capable of mitosis, okay, with only a few exceptions in the adult organism, such as cardiac and skeletal muscle and neurons in nervous tissue. Lipid components. Um, the plasma membrane is primarily composed of phospholipids, which are modified triglycerides from which one fatty acid's been stripped, and we've added a phosphate and amino group in order to impart some charge to one end of the molecule. And that creates an amphipathic situation in which the nonpolar components of the molecule associate with other nonpolar components, and the other end of the molecule, which is charged, is going to associate either with water or with cytosol. So that's why you make a phospholipid bilayer with a hydrophobic core and hydrophilic edges. Okay. In addition, you're going to find cholesterol, glycolipids throughout the membrane. Cholesterol we get from our diet. It is an unusual lipid made up of a fused ring system that serves as the starting point for steroid hormone production. And cholesterol by itself is also used to make bile salts and to stabilize membrane fluidity. The phospholipid is the major building block of the cell membrane, and the glycolipids are designed to, in part, identify the cell as belonging to the body so that the immune system is able to tolerate it. This is just a little rundown of phospholipids in the bilayer. Okay. This forms a semi-permeable barrier which allows only certain things through on their own, right? Nonpolar components can go directly through the cell membrane without any trouble. Examples: oxygen, steroid hormones, lipid-soluble drugs and vitamins. But anything polar or charged is going to have to pass through a gate, a tunnel, or a pump in order to get from one side of the membrane to the other. And so protein is usually the material from which those are made. And the cell can determine how many of them there are in the membrane and whether they're open or closed. A little discussion about cholesterol here, how it works, and glycolipids, right? The glycocalyx is just the outer sugar covering around the cell that serves as part of the extracellular matrix, but also identifies the cell surface to the immune system. And it's located only in the region of the phospholipid bilayer that faces the extracellular material. So you can see here what we call the fluid mosaic model, where you've got different molecules here that are throughout the membrane, and there is some degree of movement either left or right or forward and back. There are cytoskeletal components in many cases that are connected to the proteins that are embedded in the membrane, or to peripheral proteins which are on one or the other side of the membrane. And then you've got cholesterol, you can see in yellow, phospholipids, and then you've got the carbohydrate component, which can be attached either to protein or to lipid. Membrane proteins make up half of the plasma membrane by weight. They float and move in the fluid bilayer. Again, the degree of movement depends on the nature of the protein. Some are going to move quite a bit, okay, drift throughout the membrane. Some are going to be relatively fixed in their location because of their attachment to the cytoskeleton or their presence on relatively immobile parts of the plasma membrane called lipid rafts. And they perform many of the membrane's functions. The integral proteins pass all the way through the phospholipid bilayer from one side to the other. So many of these are gates, pumps, or tunnels, while the peripheral proteins are either on the cytoplasmic or the extracellular side of the phospholipid bilayer. And so that explains the location of these types of proteins. Note that for either peripheral or integral proteins, the amino acid residues are going to reflect the location of that portion of the protein, right? If it's an integral protein, the amino acid residues that are in contact with the hydrophobic tails of the phospholipid are going to be of the nonpolar variety, okay? Whereas the core of the integral protein, if it's, say, a tunnel or a pump or a gate, is going to be coated with either charged or polar amino acid residues. And again, that that reflects the low energy arrangement of that particular protein with regard to A, its shape, and B, its association with its surroundings. Remember that like dissolves like, right? Nonpolar material tends to associate with nonpolar material, and polar material tends to associate with polar material because of that second law of thermodynamics. It states that in any system we consider, we try to find the low energy state. Okay? Transport proteins regulate movement of substances across membrane. They include channels, carriers, pumps, symporters, and antiporters. An antiporter moves two components in opposite directions across the membrane, while a symporter moves two components in the same direction. The ligands are what bind cell surface proteins called receptors. Examples include neurotransmitters that can bind to muscle cell receptors to initiate contraction by opening a ligand-gated ion channel. Hormones can do similar things. Okay, they can bind ligands and initiate responses in the target cell. Identity markers communicate to other cells that these cells belong in the body and shouldn't be destroyed, so you can think of them like chemical ID tags. Other roles, enzymes can be on the cell surface, either facing the extracellular matrix, or it can be on the cytoplasmic side, and they simply lower the activation energy of the chemical reaction so that it happens more quickly. And anchoring sites can secure the cytoskeleton to the plasma membrane, while adhesion proteins lock these cells either to the extracellular environment or to each other. And there's examples of each type. Okay, so those are some of the components we've gone through. Membrane transport is just a mechanism to move material across the cell membrane either with or against its concentration gradient. If we're moving with the concentration gradient, we're going to involve passive transport processes such as diffusion, facilitated diffusion, and osmosis. While if we're moving something against its concentration gradient, we're going to use an active process such as primary or secondary active transport or vesicle-mediated transport. Okay? So the cell needs to do both of these things. So you can see here that the cell determines what it wants to bring in or leave out, and the cell also determines which direction it goes in, and also how quickly that material moves in or out by controlling either the number of these transport proteins in the membrane or whether they're in an active or an inactive state. Okay? So the cell gets to determine what it wants to bring in and bring out. Membrane transport passive processes don't require any energy. Diffusion and osmosis work with the concentration gradient from high to low concentration. Diffusion refers to the concentration gradient of the solute from high to low, and osmosis refers to the concentration gradient of water from high to low. Active processes in membrane transport require energy. The substance has to be moved up its concentration gradient, or in vesicle-mediated transport, the vesicle has to move within the cell and either spit something out or take something in, or in some cases do both. And that requires energy-mediated interaction with cytoskeletal components in order to promote the movement. So there's diffusion, right? Which is the movement, the passive movement of solutes from high to low concentration. Eventually we reach a condition called equilibrium, in which there's no net movement of the solute. That doesn't mean there's no movement. It just means that movement in direction A is occurring at the same rate as movement in direction B. Okay? So the molecules get evenly distributed. The rate of diffusion depends on several factors: the steepness of the gradient, the temperature of the system, and the size of the thing that's diffusing. Okay? The steeper the gradient, the more rapid the diffusion. The higher the temperature, the more rapid the diffusion. And the smaller the item diffusing, the more rapid the diffusing. And you can see examples again of this here. Okay? That's simple diffusion. Small and nonpolar solutes do this in either a gas or a liquid. Okay? So they move with their concentration gradient from high to low without you having to do anything about it. This is not regulated by any property of the plasma membrane. For things that are non-polar, all right. For things that are polar, this can be regulated by the state of leak channels or gated channels in the membrane that allow material to move with its concentration gradient from high to low. It continues to this movement as long as our gradient is existing. Once the gradient vanishes, then there's no net movement. So you can see examples here of simple diffusion, right? This happens without proteins for nonpolar material. Polar material has to go through with the help of a channel, and that's why we call it facilitated diffusion, okay? So anything polar or charged has to have a special hole to go through the phospholipid bilayer, and that's usually a protein tunnel or pump, or not a pump, but a protein tunnel or a gated tunnel. That's facilitated diffusion, right? The two types are channel and carrier mediated. In channel mediated diffusion, you have a tunnel that goes through from one end to the other. In carrier mediated diffusion, we pick up something on one side of the membrane, move across the membrane, and dump it on the other side. Okay, so that's channel mediated. If it's a leak channel, the tunnel is always open. If it's a gated channel, then whether it's open depends on what that gate responds to. Okay, some gates respond to some sort of physical force. Some gates respond to a change in voltage, and some gates respond to the binding of a particular ligand, okay, chemical that holds the door open or slams it shut. Okay, so you can see again back here with simple diffusion. Okay, that's carrier-mediated diffusion. Polar molecules are aided by a carrier protein. They bind the substance on one side of the cell membrane, they shuttle it across, and they dump it on the other side. Okay, if it's a uniporter, the carrier transports one substance in one direction. If it's an antiporter, two substances in opposite directions. And if it's a symporter, two substances in the same direction. Okay? So you see an example here of a uniporter, right? Glucose in the interstitial fluid is moved through the plasma membrane with a carrier protein and dumped on the other side. Hush. I love you too. Okay, you got to be quiet. Daddy's talking. Okay. And so what you're seeing here, again, is movement of material across the hydrophobic core that couldn't take place without the aid of this special carrier, right? Because the glucose couldn't pass through those fatty acid tails, right? That energy barrier is too large. And again, this is working with the concentration gradient in this case. Okay? Osmosis is simply the movement of water down its concentration gradient. So essentially the way to think about this is that water chases solutes. So water is going to go where the solute concentration is the highest. Okay? Passive movement of water through a selectively permeable membrane can occur because of water's small size. All right? But most solutes will not pass through. There are also water channels called aquaporins, which are protein channels, that can increase the water permeability of the cell membrane, and the cell can determine how many of those are produced in order to adjust the rate of osmosis. Okay? So those are the two ways that we cross right? The solutes that can move across include permeable solutes that can pass right through the bilayer, and those are either small or mostly nonpolar material: oxygen, CO2, urea, um, nitrogen, water, all examples of this. Non-permeable can't go through the bilayer because they're charged or polar. Examples given: ions, glucose, proteins. Concentration gradients across the plasma membrane determine the rate at which this occurs. It causes water concentrations to differ on one side of the membrane or the other. The greater concentration of solute is going to pull the water towards it. Movement by osmosis is dependent on the gradient between the cytosol and the solution. The water will move down the gradient until an equilibrium is reached. Okay, so um, essentially, this will reach equilibrium when forces driving the opposite movement are equal to the forces of osmosis. Okay, so osmosis can drive, let's say, fluid into the cell until hydrostatic pressure in the opposite direction balances out the osmotic pressure, and then no net movement occurs. Right? You can see examples here, right? Osmosis in the cell being driven by differences in solute concentration and the steepness of the gradient. You see the aquaporins at work as well. Osmotic pressure is just the pressure exerted by the movement of water across a semi-permeable barrier such as a cell membrane. Um, Again, the hydrostatic pressure comes into play once we reach equilibrium. And so you can see here an example of an experiment you can do. On one side of a semi-permeable barrier, we have a solution, and on the other side of the semi-permeable barrier, we have a less concentrated solution, or we could even have distilled water, right? The water can pass through the membrane, the solute cannot, and so what happens during the course of the experiment is that the water chases the solutes on the other side of the barrier, the fluid level goes up, and then eventually we reach equilibrium when the hydrostatic pressure in the opposite direction is the same as the osmotic pressure that's pulling the fluid across the membrane from right to left. A cell gains or loses water with osmosis along a change in the cell volume. Okay, the tonicity is just the ability of the solution to change the volume of the cell, either by shrinking it or growing it. And the three types of tonicities are isotonic, hypotonic, and hypertonic. In an isotonic situation, the water concentration on either side of the cell membrane is the same, and so there's no net movement of water, and the cell retains its function. In a hypotonic situation, the water concentration outside the cell is greater than inside. Water moves into the cell at a greater rate than it moves out. The result is the cell swells and can eventually rupture. In a hypertonic situation, the water concentration inside the cell is greater than outside. Water leaves the cell quicker than it goes in, and the cell shrivels up, and that's a process known as crenation. In both hypotonic and hypertonic situations, the cell is going to lose function. Okay, and so in the body, we work really hard to try and maintain isotonic conditions, and we do that through the action of the integumentary system, the urinary system, the digestive system to some extent. Okay, all sort of work together to do this. So there's isotonic, there's hypotonic. Okay, an example of a hypotonic effect on the cell: it swells up and it ruptures. That's called lysis. If this happens to a red blood cell, it's called hemolysis, which is just lysis of red blood cells. And there's hypertonic with crenation. Okay, so this is why when we give a transfusion, if we need to introduce fluids back into you because you're dehydrated and you can't take anything by mouth, we never give you distilled water, because you would end up rupturing the red blood cells, releasing the hemoglobin inside them. Those would clog capillaries, and the result would be the death of the individual due to lack of blood flow to anything downstream of where the blockages would occur. So you can see again what happens in the three different situations. Okay. Active processes include primary and secondary active transport and vesicle-mediated transport. In primary active transport, we use ATP as fuel to move a solute against its concentration gradient either into or out of the cell. Okay? So this is basically a protein pump driven by ATP. In secondary active transport, we utilize a gradient produced by primary active transport to force material either into or out of the cell against its concentration gradient. And then in vesicle-mediated transport, we either pull material into the cell in a membrane-enclosed vesicle, or we kick material out of the cell by moving a membrane-enclosed vesicle to the cell membrane and causing it to fuse and evert and eject its contents into the outside environment. Okay? So that's primary active transport. Ion pumps are examples of this. Okay? Cellular protein pumps move ions across the membrane and maintain a property called membrane potential, in which the inside of the cell is more negative than the outside at the membrane location, and that can be used as a form of potential energy to drive processes that keep the cell alive, such as action potentials in nerve and muscle cells, and transport processes in other cells in the body. Okay, so these ion pumps, the sodium potassium ATPase is an example. We'll get into that in a bit. Calcium pumps in the plasma membrane of red blood cells prevent it becoming rigid from getting calcium accumulating inside, causing the membrane to stiffen. That's because calcium salts are relatively insoluble in water. There's the sodium potassium pump. It burns a single ATP, and that causes it to kick 3 sodium ions out of the cell for every 2 potassiums that it kicks in. So just by the pump running, it creates a charge separation, which contributes to the membrane potential, along with large negatively charged molecules that are locked inside the cell, most of these being proteins, and the fact that the leak channels in the cell are such that we're much leakier to potassium ions than we are to sodium ions. So the cell tends to lose positive charge to the extracellular environment quicker than it picks it up. And that's secondary active transport. We generate a concentration gradient by the action of primary active transport, and then we use that to drive material either into or out of the cell against its concentration gradients. Okay? 2 types include symports and antiports. We've discussed what those do. Okay? You can see examples here, right? This is a symport. Sodium glucose symport is going to move glucose into the cell against its gradient, while sodium moves into the cell with its gradient. And then in an antiporter, we move 2 materials across in opposite directions. Okay? So you can see here that the protons are moving against their concentration gradient out of the cell, while the sodium is diffusing down its concentration gradient and driving this antiporter activity. Vesicle transport is just the movement of materials through the cell by enclosing it in a membrane enclosed vesicle, which then interacts with cytoskeletal components to either run to the membrane and fuse and then eject the components into the outside, and that's exocytosis, or to pull material into the cell in an effort to process or destroy it, and that's endocytosis. Endocytosis comes under different categories. Endocytosis of liquid is called pinocytosis, and endocytosis of solid material, non-specific solid material, is called phagocytosis. And then there's receptor-mediated endocytosis, where we pull a particular ligand into the cell as it interacts with an area of the membrane that's coated with receptors for that ligand, and that will then move that material to some organelle in the cell where it will be processed. Okay, so those are the different vesicle-mediated modes of transport, all of which require ATP for movement. Okay, And there's how they work. Right? There's endocytosis. Okay. Functions: uptake of material for digestion, retrieval of membrane regions from exocytosis, regulation of membrane protein composition. Okay. There's phagocytosis. Purpose of phagocytosis primarily is to destroy pathogens. Okay. Um, you engulf bacteria and other pathogens through the action of phagocytic cells such as neutrophils and macrophages, and that phagosome, that membrane enclosed um, vesicle with the solid material inside, then fuses with a lysosome whose powerful lytic enzymes destroy it. And then some of those breakdown products are ejected from the cell. Some of them are displayed on the cell surface to warn the immune system that there is an infection. Okay, and some are repurposed to build cellular components. And there's phagocytosis. Okay, there's pinocytosis. There's phagocytosis. Okay, receptor mediated. Okay, one example of this is the movement of cholesterol into the cell. Right, cholesterol is a lipid, is transported through the blood um, in in vehicles called lipoproteins, which have on their surface antigens. When these lipoproteins arrive at the cell where they're going to dump their payload, they they attach to receptors on the cell surface, and that promotes receptor-mediated endocytosis of the payload inside the lipoprotein, which is some sort of lipid-soluble substance, and then the cell then utilizes that for whatever purpose it needs, and then the component that fused with the membrane is then usually ejected so that it can go eventually back to the liver and repeat the process of carrying lipid-soluble material through the blood because it doesn't dissolve very well in plasma. Okay, so there's receptor-mediated, right? If you lack the receptor for something like these lipoproteins, then you have increased risk for atherosclerosis. Okay, it's a condition called familial hypercholesteremia, and that indicates that your levels of free cholesterol and your blood triglycerides will be high because you don't have the necessary equipment to pull those lipoproteins and their payloads into the cells. It's usually inherited as an autosomal recessive, which means it's on a chromosome that contains body plan information, and you have to get two bad copies of the gene from both your mom and your dad in order to have the condition. And so that's a discussion exactly of this. Okay. Membrane potential is simply the separation of charge across the plasma membrane that can be used as stored energy to drive processes that keep the cell functioning and alive. Membrane potential is established by differential leak channels that are embedded in the membrane, more potassiums than sodium leak channels, the sodium-potassium pump activity, and the fact that large negatively charged ions are locked inside the cell and can't get out. Okay, and so that difference in charge, just like a charged battery. And when we open a channel that lets charged particles flow, that's a form of energy called current, and the result is that can be harnessed to do work. Okay, so that's resting membrane potential. Okay, there you can see when the cell is quote unquote at rest, how we get the polarization of the membrane, which is like the charge on the membrane that can be used to drive all these different processes. Okay, again, when a cell is at rest, it's not really resting; it's doing a whole bunch of work. But the reason we mean resting is because the membrane potential in this case isn't changing. Okay, and it takes a lot of work to keep it from changing. Right, 3/4 of your ATP is used just to do this, to maintain this membrane potential that keeps the cell alive, and the byproduct of that is heat, which maintains body temperature. So there is the, the major players, right? Sodium ends up more concentrated outside the cell than inside, and potassium is more concentrated inside for the reasons we've already discussed, right? The differential leaking across the membrane and the sodium-potassium pump. Right? Sodium diffuses into cells from the interstitial fluid simultaneous to the loss of potassium, which enters through sodium leak channels down its concentration gradient, pulled by an electrical gradient, and the leak channels prevent as much sodium into the neuron as potassium out, and as a result, the inside becomes more positive. Okay, so that's an example of um, what we call RMP, resting membrane potential, waiting for an event to harness this separation of charge to generate current to do work. Okay, so that's why, right, the pumps are always running, the leak channels are there, the large negative ions are locked inside. Okay. Now cells communicate with each other as well, right? Um, they have to do this in order to coordinate activity. Examples, right? Cells of the immune system distinguish normal from unwanted cells by scanning for antigen displays. The sperm and the egg um, have interaction via the glycocalyx, which allows recognition by sperm during fertilization. Cellular regrowth following injury is triggered by damaged tissue replaced by cell division in the epidermis, and cellular contact then prevents overgrowth. Right? Most cell communication occurs through ligands, which are just chemicals that attach through to a receptor or to a gate, and thus change the shape of that protein, leading to a response. The molecules that bind with macromolecules, neurotransmitters from nerve cells, hormones from endocrine tissue, control growth, reproduction, and other processes. Three types of receptors that bind ligands: channel-linked, enzymatic receptors, and G-protein coupled. Okay? In a channel-linked receptor, the ligand binds to the channel and either opens it or shuts it, and that permits flow of the thing the channel is specific for, either into or out of the cell. Okay, so examples of how this could work: ligand-gated sodium channels, for instance, in nerve and muscle cells, can initiate depolarizing events that can lead to the production of an action potential, which is a long-term electrical signal that eventually results in either a response within that cell, such as in muscle tissue, making it contract, or the release of neurotransmitter by the axon terminal of that cell on another target, causing it to potentially respond by generating an electrical signal. In enzymatic receptors, we bind a ligand and then we trigger an enzyme either on the cell surface or inside the cell that leads to a cellular cascade, which is a type of signaling mechanism that takes a small initial event and amplifies it geometrically very rapidly, and then that leads to a particular cell response. Okay? An example is a G protein receptor. The G protein gets its name from the fact that it's got a GTP bound to it, and what happens when the ligand engages the receptor is that the G protein kicks out a GDP, binds a GTP, and then activates a target. Right? It might open a channel, or it might activate an enzyme, okay, such as adenylate cyclase, and then that second messenger would cause a cascade that leads to a cellular response. Okay. Membrane-bound organelles are going to be surrounded by a phospholipid bilayer. Examples include the rough and smooth endoplasmic reticulum, the Golgi apparatus, the mitochondria, the lysosomes, the peroxisomes, and you could even argue for the nucleus. Okay. The ER are extensive interconnected membrane networks that come in two flavors: the rough ER, which is studded by ribosomes on its cytoplasmic surface, and the smooth ER, which is not. Okay, it composes about half the membrane within the cell, You can see what it looks like here. Okay. The purpose of the rough ER is to serve as a receptacle for proteins that are eventually going to either be secreted or become part of another membranous organelle, either inside it or on the outside of the membranous organelle as a membrane protein, okay, or could be incorporated into the plasma membrane or even ejected from the cell. Okay. The smooth endoplasmic reticulum is very different. The smooth endoplasmic reticulum lacks ribosomes. It's a calcium storage area. It's an area for the synthesis of cholesterol and the synthesis of lipids, okay, and the breakdown of fatty acids, and the detoxification of things like free radicals, okay, just to name a few of the processes. Okay. So you can see here the rough ER we've discussed, the smooth ER, carb metabolism as well, synthesis, transport, and storage of lipids. Okay. The Golgi apparatus is a series of flattened sacs. The cis face faces the rough ER, while the trans face faces the plasma membrane, and it's simply a distribution center for proteins that arrive from the rough endoplasmic reticulum, and it sends those proteins through secretory vesicles to their final destination in the cell, where they'll be dropped off and begin to function. Okay? You can see it here. Okay, the cis face on this side, right? The trans face on the other side. So what happens while we're in here? A process is called post-translational modification, where there are functional groups added to the protein in order to direct its future activity. These could be carbohydrates, they could be ubiquitins, they could be any number of molecules that get latched on either to the phospholipid component of the membrane or the protein component of the membrane. Lysosomes contain powerful hydrolytic enzymes that are used in recycling worn-out organelles and molecules, and also in the dissolving of bone. And also, they inject their contents when the cell decides to commit suicide in a process known as apoptosis. Okay, so this is like the stomach of the cell, and if there are any problems with lysosome function, like it doesn't do what it's supposed to, then waste products rapidly accumulate in the cell and can kill it. Okay, an example of this is Tay-Sachs disease, in which enzymes in the lysosome that break down a class of molecule called gangliosides aren't working, and the result is the accumulation of junk in the neurons, and this usually results in death before the end of infancy. And it is again an autosomal recessive disorder, right? Peroxisomes look very similar, but their function's different, right? They do chemical digestion, beta oxidation of fat, and lipid synthesis, okay? And they also have a role in detoxification. Lysosomal storage diseases such as Tay-Sachs are discussed here. Okay? The endomembrane system is just the mechanism whereby we synthesize and then distribute the proteins that are either going to be incorporated into the membrane, exocytosed, or incorporated into a membrane-bound organelle. Okay, so you can see that we move from translation on the rough ER through the rough ER as we begin to fold into our native conformation, and then transport vesicles move us to the Golgi. In the Golgi, we get functional groups added, and then we get loaded into a secretory vesicle that is then going to interact with cytoskeletal components until the payload is dropped at its final location in the cell so it can function. The mitochondria are the powerhouses of the cell, and they generate the bulk of the ATP used to do cellular work. They have two sets of phospholipid bilayer, an outer membrane, which is similar in composition to the plasma membrane, and an inner membrane, which is similar in composition to bacterial membranes. The ridges and grooves in the inner membrane provide extra surface area for the components of the electron transport chain, which make the bulk of the ATP in the cell when there's oxygen present. Okay? They have their own ribosomes and their own DNA because it's thought that they're actually descended from ancient bacteria, and their genetic material and their ribosome composition tends to reflect that. Okay, they divide on their own schedule. Cells that need more energy have more mitochondria, and the mitochondria are bigger. Okay, the ribosomes are simply the site of protein production in the cell. So you could have free ribosomes in the cytoplasm that produce proteins that stay there. Many of these are enzymes or cytoskeletal components. And then you've got ribosomes on the endoplasmic reticulum that produce the secreted proteins, or the proteins that get incorporated into membranous organelles or incorporated into the plasma membrane. And there's what the rough ER looks like. Okay. And there's the ribosome. The three sites are the E, the P, and the A site, and they essentially read the codons in the messenger RNA and incorporate the appropriate amino acid in the polypeptide chain. Okay? The A site is the aminoacyl site, the P site is the peptidyl site, and the E site is the ejection site. Okay? So we move down 3 nucleotides at a time. We read that codon, we find the transfer RNA whose anticodon matches or is complementary to the codon. It will be attached to an amino acid that is going to be part of the functional protein. And then we slide over to the next codon. We bring in another transfer RNA whose anticodon loop is complementary to that codon. We put the second amino acid connected to the first amino acid by creating a peptide bond through dehydration synthesis. We eject now the uncharged transfer RNA so that it can go back and get an amino acid attached to it through enzymatic activity. And this cycle repeats until we get all the way to the stop codon, at which point a termination factor binds the A site, the ribosome disengages from the message, the protein is released, and then the cycle can, if necessary, start over again. The centrosome are just perpendicular cylindrical centrioles and are the site of microtubule synthesis. They make the microtubules of the cytoskeleton, whose job it is to position organelles during interphase so that they function in an optimal fashion. And they're also critical in mitosis as they separate duplicated chromosomes during cell division so that each new cell has a complete DNA complement in order to cause the cells that result from the division to look and act like the original cell. So you can see what they look like here. Okay? Proteasomes are just barrel-shaped protein complexes that can digest organelles located in the cytosol and the nucleus. They degrade cell proteins through ATP-dependent pathways. Proteins get marked with ubiquitin so that they can get destroyed. With age, they may be unable to normally remove proteins, and that can lead to the death of the cell. So you can see an example of a proteasome here. Okay? Cytoskeletal components include the microfilaments, the intermediate filaments, and the microtubules. Okay? They maintain the shape of the cell. They contribute to the cell's ability to produce cell-cell junctions, and they also promote the appropriate movement of organelles and chromosomes inside the cell. Okay? The microfilaments are primarily made of actin that maintains cell shape and internal support and aid in cytokinesis, which is the division of all the other cellular components after mitosis is complete. Intermediate filaments are rigid and are designed to promote cell-cell junctions and to stabilize membrane protein position. So they're sort of like a framework for the cell. And then the microtubules are the largest of the three types of filaments, and they serve as train tracks or pulleys to move organelles or chromosomes to a particular destination. Okay? And of course, they are manufactured by the centrioles. So you can think of the microtubules as fishing line, and you could think of the centrioles as the rod and reel. Okay? And the target that's being moved would be the fish. Okay? So there are the different cytoskeletal components. The cilia are just hair-like projections that move substances across the cell surface. Usually this is the apical part of the cell. Flagella are wider than cilia and move entire cells from one location to another. The only flagellated cell in the human body is the sperm cell. And then microvilli are just membrane extensions designed to increase surface area to aid in absorption and secretion. And they have primarily a microfilament core. So you can see examples here of microvilli with the microfilaments, right? The different types of cell-cell connections include tight junctions, desmosomes, hemidesmosomes, and gap junctions. Okay? The tight junctions are rows of proteins that link cells together, preventing substances from passing between them. Examples would be, for instance, an epithelial cell of the skin, in order to cause only certain materials to be able to move through the skin. Desmosomes are attached to cytoskeletal components and latch cells to each other. Examples where desmosomes will be found will be epithelial and connective tissue, and even muscle tissue. Hemidesmosomes are half desmosomes, and their job is to attach the basal part of the cell to extracellular matrix or basal lamina, and can also have a signaling component to them. And then gap junctions are just communication networks between cells that let small particles move quickly from one cell to the next. And so you can see here how the different junctions are employed. Okay, the nucleus is where the genetic material is located. Tight junctions, desmosomes, gap junctions, we've discussed, right, what they do. Okay. The structures of the nuclear envelope and the nucleolus. The nuclear envelope is a double phospholipid bilayer. The outer phospholipid bilayer is continuous with the rough endoplasmic reticulum, while the inner phospholipid bilayer is attached to scaffolding proteins that maintain the shape and the integrity of the chromatin. Okay? Nuclear pores are just large tunnels through the nuclear membrane that allow the movement of big molecules either into or out of the nucleus. And the reason you need these is to get assembled ribosomes out of the nucleus from the nucleolus, and to get ribosomal proteins into the nucleus to eventually make it to the nucleolus, and then to allow proteins that regulate transcription to move into the nucleus from the cytoplasm, okay, and also allow messages to escape. So those are big molecules, and so they need big holes in order to get from one side to the other. The nucleolus is just a site of transcription of ribosomal RNA, and it serves as an area of ribosomal assembly. Okay, this is where ribosomal proteins attach to ribosomal RNA to make a functioning ribosome, which can then exit through the nuclear pore and then take up residence on the endoplasmic reticulum or remain in the cytoplasm and begin the job of translation. Okay, it's usually a dark staining region in the middle of the nucleus and is attached to what are called nucleolar organizing regions, which are simply the ribosomal RNA genes. So you can see the nucleus and the nuclear membrane here with the pores and the chromatin and the nucleolus. Okay. DNA is housed in the nucleus and is composed of nucleotides, and it has a sugar phosphate backbone, and the core of the DNA double helix is made up of nitrogenous bases, okay, which are either single or double ringed organic components that contain the information portion of the molecule. Each deoxyribonucleotide is made up of the 5-carbon sugar deoxyribose, phosphates, and one of 4 nitrogenous bases, either adenine, cytosine, guanine, or thymine. Okay? The nucleotides are linked by phosphodiester bonds. Each molecule has two complementary strands of nucleotides, which means that if on one strand of the DNA there's an A, an adenine, on the opposite strand at the same position, there's going to be a thymine. And the same is true of guanine corresponding to cytosine. And the reason that that's important is because that rule of complementarity is what guides the production of new strands of DNA when the DNA reproduces. When this happens, the double helix unwinds, and then the DNA synthesizing proteins march down the old strand and incorporate the complementary nucleotides as they go in the 5' to 3' direction. And the result is the preservation of the information so that we can pass it on to new cells. Okay? Remember that the sugar phosphate backbone is going to face the cytoplasm or the nucleoplasm and is generally negatively charged, and while the core of the DNA double helix tends to be nonpolar, and the bonding that takes place between the complementary bases is the result of hydrogen bonding. Okay? Remember that humans have 46 chromosomes, 23 from your mom, 23 from your dad, and that determines the characteristics of the cells that make up the tissues, that make up the organs and organ systems that make up the body. Okay? The DNA is associated with histones and non-histone proteins. The purpose of the histones is to compress and protect the information in the DNA, and is also thought in some cases to direct transcription to some extent, while the non-histone proteins are thought to be transcriptional regulators. When DNA is not dividing, the genetic material is in the form of chromatin. When the DNA divides, an extra level of compression occurs, and we generate what are called chromosomes. The reason for this is to protect the information where we're pulling it around inside the cell. And you can see here in this diagram how the complementarity works, right? G pairing with C and A pairing with T, all ways to preserve the information. In the event that synthesis ends up not working the way we anticipate, and there's a goof, we call that a mutation, right? And mutations can have one of three consequences. They could be good, which means adaptive, so they produce a characteristic that increases the likelihood that organism will live long enough to breed and pass its genetic information to its offspring. They could be bad, which is non-adaptive, which means to be less likely to live long enough to breed, or they might not have any effect. Okay? Mutations come from mutagens, which are chemicals that attach to DNA and change its shape. It can come from radiation. They can also come from viruses, and also errors in the DNA synthesis process, which is not perfect. Okay? Genes are just information blocks of DNA that contain protein building instructions. The anatomy of a gene begins at the start of the gene, the 5' end, where a promoter is the gatekeeper to the rest of the gene, and that promoter is used to assemble transcription factors and RNA polymerase at the beginning of the gene. The exons are stretches within the gene that contain protein building information, and the introns are regions of DNA within the gene that contain what is thought mostly to be regulatory information. Okay? The nucleus and the ribosomes coordinate the process of transcription and translation. Okay? Transcription is the process of making an RNA copy of a particular gene. That RNA copy is made beginning after the promoter and ending at the transcription termination site. Once that RNA molecule is produced, it has to be processed so that it can be used for translation. To do that, we have to remove the intron components from that RNA molecule. We have to add a chemical cap to the 5' end to mark the beginning of that molecule, and then we have to put a poly A tail on the end of that molecule, and that is a mature messenger RNA which can then leave the nucleus through a nuclear pore, and then ribosomes can bind it in the cytoplasm or in the rough endoplasmic reticulum, and we can begin the process of translation. which is the use of that genetic information in order to generate a protein that the cell needs. Okay? So what happens in translation is that the ribosome binds and scans the beginning of the messenger RNA, looking for a special codon called an AUG. Right? Once it finds the appropriate AUG, it will pause, and then the ribosome will bind to its first transfer RNA that has an amino acid attached to it. The fit of the anticodon loop in the transfer RNA and the codon in the message will be tested. If the fit is good, that amino acid will be incorporated into the protein. The ribosome will then move down 3 more bases. Okay, the now open A site will pick up another transfer RNA with the appropriate amino acid for the codon that the A site is sitting over. The discharged transfer RNA in the E site will be ejected, and a peptide bond will be formed between the amino group of amino acid 1 and the carboxyl group of amino acid 2, and the result then is that the protein has now increased in size by 1 amino acid. And this cycle repeats until we get all the way to the stop codon, which causes a termination factor to bind to the A site of the ribosome. That results in release of the protein and the disassembly of the ribosome, and then the process can start over if we haven't yet fulfilled the quota for that particular protein. Okay? Ribosome is just the temporary copy of the genetic material in the form of messenger RNA. It contains ribose as the pentose sugar instead of deoxyribose. Other important RNAs include ribosomal RNA, which is a structural component of the ribosome, and transfer RNA, which is the molecule that brings amino acids to the complex of the ribosome and the message in order to build the protein. Okay? The 3 events of transcription: initiation, elongation, and termination. In initiation, the DNA is unwound after assembly of RNA polymerase and transcription proteins on the promoter. Okay, the DNA strand is copied. This is the template strand. The other strand, which is the coding strand, is not copied. In elongation, free ribonucleotides are base paired. We make an RNA copy of the nascent gene. Okay. And then in termination, the RNA polymerase is released at the end of the gene. Hydrogen bonds are broken between the DNA and the new message, and the DNA rewinds into a helix, and then we process out the introns, we cap and polyadenylate the message, and then it can leave through the nuclear pore. Okay, and so that's transcription. That happens in the nucleus, right? Those are the modifications, right? The exons and the splicing, okay? The capping, okay? The poly A tail. The reason for the cap and the poly A tail is to identify the 2 ends of the message. The tail kind of acts like a clock, right? The longer the tail, the longer the message is going to spend in the cytoplasm. The shorter the tail, the less time it's going to spend there. Once the message has served its purpose, we can break it down and recycle its components. Okay? There's translation, right? Transfer RNA, messenger RNA, all part of this, right? The A, the P, and the E site, right? The mRNA is organized into codons, right? With a start codon, the AUG, and the stop codon, one of 4 different stop codons where our messenger RNA translation stops, and the transfer RNAs which bring the amino acids to the growing protein, right? Aminoacyl tRNA synthase is what generates charged transfer RNAs. It's just a cytoplasmic enzyme, okay? And again, this requires ATP to go, okay? Those are the 3 sites, E, P, and A, right? There's the message with the codons stitched together, and you can see the transfer RNA with the anticodon loop. That's what's going to sit on top of the codon, and if the base pairing is good, then that amino acid gets incorporated, right? So we build it one peptide bond at a time, the information in the gene directing the assembly of the protein, right? And then the protein will begin to fold into its functional shape, directed primarily by chemical interactions within the chain and chemical interactions between chains. Okay? You can see the genetic code here. Okay? That's which codon in the message is going to describe which amino acid gets incorporated into the protein. Right? Initiation, elongation, and termination, the three processes of building the, the protein. Right? In initiation, everything assembles at the AUG that marks the start of the open reading frame, which is the stretch of the messenger RNA that contains the protein building instructions. Okay. Um, Elongation, right? The tRNAs come in, they dock on the A site. The amino acid in the A site gets attached to the amino acid in the P site, and then we slide down 3 and we kick out that now empty transfer RNA, and we do that until we get to the stop codon where the release factor binds the A site, the ribosomal subunits split, and the protein's released. Okay. Sometimes you get multiple ribosomes doing this, so we call that a polyribosome. Okay, as we march from one end to the other. Okay? DNA guides all the cell's activities, right? It's responsible for all the enzymes that produce the chemicals needed for the cell to survive, and the proteins that maintain the cell's shape, define its membrane function, define all the functions in the cytoplasm, responsible for cell-cell junction and cytoskeletal characteristics, okay, and also define um, the role of the cell in the body, whether it's a muscle cell, a nerve cell, epithelial cell, and so forth, okay, and that's because of the use of the genetic material in the nucleus. Now, at some point, a cell will accumulate enough organelles and enough fuel to undergo cell division. Okay? There's two types of cell division: mitosis, in which you make a clone of the original cell, so you get two new cells from one original, and meiosis, in which you generate a cell with half the amount of genetic material as the original, and that's designed to produce gametes, sperm in the male and eggs in the female. Okay? In somatic cell division, it's just mitosis. In the gonads, it's meiosis. Okay? The centrosome are just the cylindrical structures that organize the microtubules that promote the chromosomal movement, forms the DNA in the cell. The nucleus, containing 46 DNA molecules, goes from chromatin to chromosomes during the process of prophase. The microtubules attach to the constricted mid portion of the chromosome called the centromere, okay, and then the microtubules, which are on opposite sides of the centromere, will pull the duplicated chromosome in half. Okay, the cell cycle is made up of interphase and the mitotic phase. Again, depending on the tissue type, these phases last different lengths of time. In rapidly dividing tissue, interphase about 23 hours, while mitosis lasts about an hour. Okay, in tissue that rarely, if ever, divides, interphase can last as long as the entire lifetime of the cell. Okay, like in a nerve cell. Generally, in most cells, mitosis, which is the division of the nuclear material, is followed by cytokinesis, which is the division of the remaining cell components. In some cells, that's not true. An example would be megakaryocytes that produce platelets in the blood. They undergo mitosis but not cytokinesis, and hence they have huge nuclei. Okay, but in most cells, it's mitosis followed by cytokinesis. The S phase of interphase is where DNA synthesis takes place. The G1 phase is growth and preparation for DNA reproduction, and G2 is growth and preparation for mitosis. Okay? So those are the different parts of the cell cycle: G1, S, and G2. Okay? Those are the steps, right? In DNA replication, we unwind the DNA molecule at the origin of replication. DNA polymerase 3 binds there. To do that, gyrase does the unwinding of the DNA molecule, while the DNA polymerase does the synthesis of the new strands. This is ATP. Well, it's nucleotide powered. The building blocks of DNA are nucleotide triphosphates, which basically have the same energy content as an ATP molecule, because they are deoxyribonucleotide triphosphates. So the power in the production of nucleic acids is contained within the building blocks themselves. At the origin of replication, the two strands separate. One strand is built continuously in the 5 to 3' direction and is called the leading strand, while the other strand is built in small segments in the 3 to 5' direction. I mean, not really in the 3 to 5' direction. It's a 5 to 3' direction, but it's done discontinuously. And then the little segments are joined through a process of replacement of little stretches of RNA used as primers to build DNA with DNA, and then ligation of those segments together to form one continuous complementary lagging strand. Okay, so you can see the process here. Okay, again, continuous on one strand, discontinuous on the other. Okay. G2 phase is brief. Centriole replication completes. Enzymes for cell division are whipped up. Here we are in interphase. Then mitosis follows the acronym PMAT, right? Prophase, metaphase, anaphase, telophase. In prophase, the chromosomes condense, the nuclear membrane disappears. The spindle apparatus begins to form and attach to the centromeres of the chromosomes. In metaphase, the chromosomes line up approximately in the middle of the cell along an imaginary structure called the metaphase plate, with microtubules attached to opposite ends of the centromere. And then at anaphase, the duplicated chromosomes are pulled apart, driven again by shortening of the microtubules, which is driven by ATP utilization at the centrioles. And the idea here is, again, to get one copy of the genetic material to each side of the cell so it can be incorporated in a new nucleus. Okay? In telophase, the genetic material arrives at opposite ends of the cell, and then we reform the nuclear envelope, and then cytokinesis divides the remaining cellular components. Production of a cleavage furrow is triggered by, again, microfilament proteins. Division of the cell contents takes place, and now we have two new cells that look and act just like the original cell. Okay? Now, at some point, the cell will reach a time when it's no longer fully functional, and it will die. Okay? The reasons for this are a little bit elusive. There's a number of theories out there: accumulated cell damage due to interaction with free radicals, autoimmune malfunction, but the most widely accepted theory is that the chromosomes lose genetic information at the tips of the chromosomes, called the telomeres. After many cell cycles, you can't complete the ends of the chromosomes effectively because the enzyme called telomerase can't keep up with the production of the new material, and eventually you lose critical information that keeps the cell fully functional, and then the cell sends a signal to its lysosomes to essentially release their enzymes, and then the cell digests itself from the inside out and it basically shrivels up and disappears. Okay? With aging, the rate of cell death outstrips the rate of cell reproduction, and the result is that you lose tissue and organ function, and eventually the organ system shut down and the individual dies. Okay? Supporting evidence for this comes from experiments where we've generated human tissue cultures that overexpress the enzyme telomerase, and what we find is that as long as we continue to feed those cells oxygen and nutrients, they don't die. Okay? Apoptosis is an orderly, programmed cell death. It's initiated by ligand receptor signaling. Okay. Actions initiated include destruction of DNA polymerase, breakdown of DNA, destruction of the cytoskeleton, condensation of the cytosol, and destruction of the organelles, initiation of signals that stimulate the destruction of the cell. Programmed cell death occurs to promote proper development during the formation of a new human being, and also to remove cells that are no longer able to function. Okay? In cancer, what happens is that the regulation of the cell cycle is ruined as a result usually of mutation. And the result is that you generate a cell that really serves no purpose to the body, but consumes oxygen and nutrients from the blood supply and invades and destroys healthy tissue. And the result is that you start to lose tissue function as the cancer spreads from its initial point through the circulatory system and the lymphatic system, and eventually you do this to so many tissues that the host dies. Okay, the approaches to treating cancer include radiation, chemo, and immunotherapy. In radiation therapy, you damage the cancer cell DNA so much that the cancer cell dies. In chemotherapy, the chemotherapeutic agents bind to the DNA of the cancer cell and ruin it, and then the cancer cell dies. And then in immunotherapy, you try to tag the tumor tissue with an antigen that the immune system recognizes and can move in and then destroy the cancer cell. Okay? None of these approaches are foolproof because cancer is really more of a symptom than it is a disease. The symptom is uncontrolled cell growth. The cause is a mutation somewhere in some cell cycle component. And so the number of genes that are responsible for regulating that are in the thousands. And so in order to effectively wipe out cancer, what we would need in the long run is an approach that looks at the initial event that produced that cancer tissue, and then we'd have to come up with some way to reverse that event, or isolate all those cells that are descended from that and cause them to commit suicide at the same time. Okay, and we just aren't there yet. We just don't have it yet. So, with that, we come to the end of this chapter. Review this. It will be on the exam this week. And if you have any questions, make sure and let me know. And I'll see you guys in class. Thank you very much.
Free at Furniture Fair. Today's the day, Cincinnati, to try something new to make your mornings better.
How long the sunshine will last?